Dr. Ben Park really doesn't even need an introduction at this point to our case conference, but he directs personal medicine initiatives associated with cancers at our cancer center. Also a world-renowned breast medical oncologist, has been in this personalized medicine space for a long time. He leads our molecular tumor boards, which he's really, brought together since his arrival, I think in 2018 at Vanderbilt, where we all come together as a group to discuss some of the sequencing results from Tumor. So again, he's done a lot in this space, even in his short tenure at Vanderbilt. Both Dr. Wiesner, myself, and Katie Lang and our other genetic counselors interface a lot with Ben. We work together on many of these cases. So welcome, Ben. Thank you for your willingness to present, and we're looking forward to what you have to say. We can't hear you.
Sorry, I'm muted. I'm not used to not having Zoom. Sorry. You can hear me now?
Yes.
Okay. I gave this talk last year, so again, I was updating because Brenda was kind enough to send me the old slides and realized that there wasn't a huge amount to update, but these financial disclosures. I also realized I should have updated that picture because I think that picture is now like 7 years old. So you can probably tell I've got a lot more gray hair. Could I get the next slide, please? So today I thought I'd talk a little bit about, I know time is limited, cancer really is a genetic disease, and I know many on this audience view this as heritable forms, which germline mutations and variants, pathogenic variants certainly exist that lead to predisposition, but really all cancers at some level, even non-heritable forms, are a disease of DNA gone bad. And as Tua mentioned, precision oncology is something that's relatively new in the field of oncology, but it really is an old idea. Can we figure out what are the genetic underpinnings that make a cancer or tumor tick so that we could potentially do something about it in terms of drugs, and also for what you all do in the germline space, strategies for risk reduction, prophylactics, etc. The problem with this has been, and again, I'm preaching to the choir, interpretation is much more difficult than I think people realize, or at least initially realize, and that goes for both the germline and somatic space. And the two are ever more convergent, meaning that germline findings are leaking over into the therapeutic space, and somatic findings are also going to have implications for families who have germline alterations in terms of what could be druggable in the future. So that's what really led to the creation of molecular tumor boards. And then some conclusions and thoughts, and hopefully time for a couple questions. Next slide. So precision oncology, what I do as a medical breast oncologist is treat patients in a curative paradigm as well as in metastatic disease, which is not curable. But in the curable paradigm, most of our drug choices and therapies are derived from large clinical trials, which are the outcomes of thousands of patients. And so we take whatever worked best for the whole of the group, and that becomes the standard of care. What that has led to is great, reductions in morbidity and mortality of breast cancer, but it also means that every stage 2 ER positive or HER2 positive breast cancer is treated almost identically because there is a standard of care for a curative paradigm. What we now know, though, with sequencing is that everyone's cancer is as different as the people who get them. So individualized types of therapies and how we do that is really what precision oncology is. At least that's kind of how I view it. And we'd like to, in the future, do this for all cancer patients, but for right now, we're doing mostly metastatic disease, where even though there are standards of care, because it's not generally a curative paradigm, there's more wiggle room to actually intervene where we find something in the patient's DNA. So how do we do this? Next slide, please. Well, as I mentioned earlier, cancer is really a disease of DNA gone bad, just whether it's heritable or not. And this diagram is kind of taken from a classic of Peter Knoll, who was the first really to associate a genetic hit that was consistent with a given type of cancer, in this case, chronic myelogenous leukemia, or CML. And he was the person who discovered what's now known as the Philadelphia chromosome, later discovered by Janet Raleigh via translocation of 9 and 22 of BCR-enabled genes. But the idea is that in the green, you have these normal cells, and there's a lot of them. one of those cells gets a genetic damage or hit. That could be a mutation, a deletion, an amplification, etc. But it leads to also a wave of clonal expansion. It really becomes a numbers game, because kind of the idea that a cancer cell is the accumulation of 3 to 8 genetic hits is true, but it doesn't really get to the heart of the fact that you need clonal expansion at every step. Meaning that now the blue cells, if it was one cell that was blue and one cell that was brown and then the red cell is cancer, but there was no expansion, mathematically that would never happen. And so you have to have at every stage some sort of proliferative event or something that lets the population grow and continue to hang around. This is also why we think most cancers happen in adults and usually in the 5th, 6th, 7th decade of life, because it takes that long to accumulate all those genetic hits. Next slide, please. Germline testing is really the first hit, as I think everyone in this audience knows, and that's heritable. And so every single cell that has a diploid complement, in theory, would have a mutation if it was inherited from either mom or dad's side. So classic examples are BRCA1 and 2. Again, I'm going to kind of skip over this since most of this is known to this audience, but we have now evolved to next-gen sequencing, and panel gene testing has become routine. Typically, though, I remind patients that this is still a relatively small number of genes. When we think about the body having 20,000 genes, and we're doing maybe at most 100 for germline testing right now, that again kind of gives you the scope or magnitude of how few genes we really test. Next slide, please. And what's been really exciting in the field of oncology is that even though in the past 2.5 decades now we've had targeted therapies, we've really only started to recently have targeted therapies that have been derived from germline or heritable gene mutations. Classic examples now are BRCA1 and 2, and probably also PARP2 with PARP inhibitors, and then also with DNA mismatch repair genes that lead to tons of mistakes in the cancer genome. Any one of those could potentially be a neoantigen that was going to respond to immunotherapies, either anti-PD-1 or anti-PD-L1. And there are probably going to be more in the future. Next slide, please. For those of you who don't know, this is how PARP inhibitors work. I always find this fascinating, and it really is a poster child of how scientific research at the bench can lead to real impact in the clinic. This was really developed by Alan Ashworth when he was at the UK, and he's now the Cancer Center Director at UCSF. But our normal cells are always having breaks in DNA, double-stranded, single-strand, and if we don't fix them, the cell has to die. And so we've evolved 2 redundant pathways to repair them: homologous recombination, which is mediated by BRCA proteins, and then base excision repair mediated by PARP proteins. And so cells that have a BRCA mutation, right, as a tumor suppressor, both alleles are inactivated somehow. They no longer have the BRCA or homologous recombination pathway, but they still have the PARP pathway. And so even in a cancer cell, if you have too many of these breaks, it's going to die. And so PARP rescues that, even in the absence of BRCA genes. Now, on the other hand, you take cells from A and put PARP inhibitors on it. This would be the normal cells of a BRCA carrier, let's say, because even though they have one heritable mutation, they still have a good copy. So they have an intact homologous recombination pathway. You poison the PARP pathway, but the normal cells are still okay because they have that BRCA pathway repair intact. On the other hand, cancer cells in a BRCA patient, usually, not always, usually have started because they've lost that second allele. So they're void of BRCA proteins, just like this in the B panel. Now the difference is you give them a PARP inhibitor, and this is called synthetic lethality, and a concept borrowed by Fly researchers. This leads to the fact that they can no longer repair the double-strand breaks and ultimately cell death. Next slide, please. Now, tumor-only testing is something that was the norm when all this business started, largely with Foundation Medicine in terms of a company. And in the past, that was relatively easy. I remember the original Foundation Medicine test, I think only had like 150 or 200 genes. And a lot of this was just focused on hotspot regions or pathogenic variants that occur with such frequency, we don't have to worry about, is this a VUS, is it pathogenic? So it was relatively easy. We've now evolved into larger cancer gene panels with several hundred genes, and whole exome sequencing is probably on the horizon at some point. But the problem with this is, as we grew the panel of genes, distinguishing between true somatic alterations could be problematic, as well as whether or not these were of consequence. Are these what we call passenger genes instead of driver, or passenger mutations instead of driver mutations? The thing about tumor-only testing, and again, many of you are aware of this, doesn't really require consent if you're just going to do tumor-only testing. Next slide, please. And so there is utility for some of the mutations with approved therapies. There hasn't been really a great randomized controlled trial that would show proof of benefit. There was one that tried this and didn't really show proof of benefit. But I would argue also that that trial is flawed by design, and any trial really is if you're just trying to compare all patients with all different types of cancers, looking at all different types of genetic alterations that got targeted therapies. There are such subtle nuances and differences between cancer types, mutation types, and even the drugs, targeted therapies that we use. Some really, I would call, not even are that targeted and have probably the same type of toxicities as chemotherapy, if not worse. So the design of trials in the past have also not been optimal. And as I mentioned earlier, most of this is being performed in metastatic disease where we don't have, for solid tumors, for the most part, curative paradigm. So it allows some flexibility and leeway in order to treat these patients. Next slide, please. So germline filtering of tumor sequencing became increasingly important. To Memorial Sloan Kettering's credit, they do their own in-house tumor sequencing, but they actually have done germline filtering from the very beginning. And I think that that was a smart thing to do because it really allowed us to get rid of the variants and determine what is really somatic and what is really germline. The advantages are that you could really see just the tumor somatic mutations. The disadvantages, which I think a lot of people did not think about at the time, is that it potentially filters out meaningful germline variants, meaning that since all of this is done electronically, there was no consent. All you had to do was just get the patient's paired normal DNA, grind it up, do the same sequencing. The computer subtracts it out. And so it never spits out as a result a BRCA1 pathogenic germline because it's just buried away in the filtering. And that is a potential opportunity loss, as we all know. Next slide, please. So if these patients are not consented, they'll never see it. That still exists today. Some tumor-only tests now state the possibility of germline variants. That still exists today too with Foundation Medicine. They'll explicitly say if they see a germline pathogenic variant in the right appropriate clinical context, genetic counseling and testing may be warranted. And then some germline testing or filtering tests selectively unfilter. I think the next slide has an example of this. Yeah, so this is a company that I'd worked with when I was at Hopkins. It's not one of the bigger ones, but they were doing germline filtering also from the very beginning. And I mentioned to them that, hey, you know, you're potentially missing things that are true germline, and they hadn't thought about that. So they went back in their data, and these were electronically the 4 genes that were most frequently filtered out with pathogenic variants. So they decided to kind of make a hybrid test where, like Foundation, they weren't going to do germline filtering on these 4 genes. I don't know what their latest, or even if the company's around anymore, but what their latest iteration of this was many years ago. But this kind of also was their kind of disclaimer that they weren't filtering these out, and if reports were had a pathogenic variant on any of these 4 genes, then under the appropriate clinical context, that genetic counseling and testing may be warranted. Next slide, please. So that brings us to the other point. One really needs to understand when companies offer germline testing, what's being tested and what isn't. I give 2 examples here. There was a patient who had a rare salivary gland tumor, right buccal mucosa was this tumor location source. It was a high pathologic tumor purity, so it was 75% tumor. But look at the source of normal DNA. It's saliva. And in a tumor in the mouth, and you're giving saliva, for sure you're going to get some cancer cell contamination, which is what happened in this case. I don't have the whole data to share with you. So none of that really became a reliable germline source. And there are many other examples similar to that. The tumor on the second test, the tumor location is a prostate cancer. Look at the pathologic tumor purity, though it's estimated to be about 20%. So most of this is normal DNA. Also look carefully underneath. Source of normal DNA not provided. So none of the companies mandate, including the one we're using right now called Tempus, none of them mandate that you have to send a germline sample. It helps for many reasons, but if you can't get access to it, they'll run the tumor only, and then it becomes like Foundation. It's a tumor only test. So that had implications for this particular patient and his provider who presented this to our molecular tumor board when I was at Hopkins. Next slide, please. he came to me and was very excited and said, Ben, look, there's an androgen receptor mutation. Should I give or not give a hormone therapy, enzalutamide? And I looked at this and then pointed out that there was no germline. And if you look at these allele fractions, again, everyone in this audience, I'm sure, understands this, that these are not likely to be in the tumor, particularly that AR variant. And at 100%, when the tumor purity was 20%, very, very unlikely. So really the only thing in this limited gene panel was probably the PIK3CA that was an activating mutation, and at 25%, conceivably probably is a cancer mutation. Everything else, when we looked it up, was probably just a SNP or a variant that we had really no bearing on this patient's tumor itself or therapy. Next slide, please. So these are the challenges in acting on it and why we actually started the tumor board. You know, can I believe in the mutations? Is there something missing? Who should be tested? Who should not? These are mostly per NCCN guidelines for germline testing and even now somatic testing. But these keep evolving as well. Year to year to year, I think, again, many in this audience knows that this is a moving target. How do we classify whether something's truly a driver or passenger, as I mentioned? When do we refer if we suspect germline testing, if there's not germline filtering, or if it's kind of flawed germline filtering? And how do we actually prioritize what's actually druggable and what's a resistance mutation? I'm not going to read all of these, but you get the idea that there are enough questions and subtleties of testing both tumor and germline that the great need arose in medical oncology, because most oncologists, maybe somewhat shockingly in 2021, are not trained in genetics. And so they don't really necessarily even understand the difference between somatic versus germline testing. So this has become a real problem in our field. Next slide, please. And so we created, whoops. Next slide. Maybe I might have taken one out. Sorry. We created molecular tumor boards first at Hopkins, and then I brought that over here, as Tua had mentioned. The first one was really called Big Hope for Vanderbilt Ingrid Cancer Center Hereditary and Oncology Personalized Evaluations. That meets weekly. We still do it on every Monday and Tuesday alternating, so that people in clinic on one or the other can actually present at 2 o'clock. Everyone's welcome on this call if you have interest to, actually I should say within the Vanderbilt health affiliated networks, et cetera. But that also led to the idea that we have a lot more need across the world. And so shortly thereafter, I started a virtual international molecular tumor board. So we've been using Zoom before the pandemic. I always say I should have bought stock, but that's called Our Cancer Genomes, which is a play on words for My Cancer Genome, which is a genomics tumor database here at Vanderbilt. And we've grown here. There's a bunch more. There's probably about 17 or 18 academic sites, 12 or 13 community hospitals, and about 7 international sites now. We meet on the 4th Monday of every month. There are challenges when you have people 13 hours difference, as you can well imagine, but we make it work. And so Singapore, which is one of those countries that are, I think, 13 hours ahead of us, presents some challenges, but their docs are great there and colleagues of ours. So again, this one's really open to everyone that wants to participate. Again, it's monthly, so it's not as frequent, but I'm also here to help. And so if someone really has a case that they want from even the outside, I'm really open to, if you want to email me and try to help you interpret the report. I think that might be all, Brenda, or next slide. Conclusions, yes. So hopefully, I think I've convinced you that tumor testing and germline testing can yield actionable variants. We know this for a fact. We have great drugs now to certain alterations, but it's only as good as the person or persons interpreting these tests. I've seen a lot of medical decision-making based upon whatever the companies say on there that really just aren't true or have been disproven to be true. And so one has to be very, very careful when looking at these results. Knowing what is being tested, filtered, and reported is, again, of paramount importance. And I'm still of the belief, I'd love to make myself obsolete and not have molecular tumor boards anymore. And to that end, we are also doing a kind of molecular oncology sub fellowship right now. But I think until then, we still need to have these types of molecular tumor boards, which have really grown over the past 5 or 6 years to help aid in decision making when patients get these types of results.